At the ISCT meeting, we had the opportunity to present, I think, some really exciting data with our ATA188 program. So as I mentioned, this is an off-the-shelf allogeneic T-cell therapy product that targets Epstein-Barr virus. Now, what we do with our manufacturing colleagues, who are really outstanding experts at developing this style of very unique therapy, we build a library of ATA188 that we can administer HLA-matched cells to the patients on the clinical trials. And so what we did at the ISCT meeting is we actually presented data with a full characterization of all of those lots of manufactured cells that our colleagues in manufacturing have created and made that inventory. And so what that entails is actually looking at a number of components to the manufactured drug, where we look at the T-cell receptor of that cell therapy product, where again, the T-cell receptor in these cells are really going after the EBV-infected B cells or plasma cells within the MS patient. So you really need to have specificity in that T-cell receptor for EBV. And what we're able to confirm with our sequencing analyses and so forth is that those T-cell receptors were indeed very specific for EBV antigen. And I should mention that when we create ATA188, we actually stimulate these normal healthy donor-derived T cells with 3 antigens. So these are small peptide fragments from Epstein-Barr virus, and they're called EBNA1, LMP1, and LMP2. And these are 3 antigens that have really been implicated in multiple sclerosis. So what we're looking for in the T-cell receptor is specificity for those antigens. And that's what we're able to show across all the different lots, no matter which HLA type that product is directed towards. And what we're also able to do is really look at the functional component of this product. So again, when we give ATA188, we don't want it to be an inert living drug product. We want it to go and for the cells to be active and really address and kill those pathogenic EBV-infected B cells and plasma cells. So what we also show in the ISCT abstract is expression of functional activation markers on those T cells, as well as proliferation potential of the cells. So really looking at the business end in terms of, are these functional cells that we've created through our manufacturing? And in point of fact, they're very functional, they proliferate very well, and they have great cytotoxicity as well. So I think the ISCT abstract really goes into detail with our manufacturing process and what an excellent job our manufacturing group really does to create this very unique type of product. And I will say, we spoke at the beginning about the challenges of cell therapy. Manufacturing is really a major hurdle there, where you have to have an expert group. You have to be able to create in a very repeatable way these products that have great characterization so that you can give them to patients and get a predictable result each time.